Hello students, this is Amivarya, Assistant Professor of Microbiology from the Department of Microbiology at LJ Institute of Applied Sciences, LJ campus. Welcome to the world of basic microbiome. Here we are going to discuss the topic normal growth curve. The study of normal growth curve is important for knowing the bacterial growth kinetics as well as the cell division methods of bacteria. This is most important from both point of view research as well as industrial. Certain species of bacteria, they are able to degrade the industrial waste easily. By knowing that species which are able to degrade the industrial waste, that is more important for modification of genetical modification and genetic engineering. That particular type of species, they can also able to grow in the oil spills, and they are able to grow in presence of certain complex hydrocarbons. By degrading that complex hydrocarbons, this type of bacteria, they can easily growing in that polluted oil spill environment, right? The study of slope as well as growth curve, that is important by two point of view. That type of bacteria, they are able to grow in the industrial waste in presence of mixture of different industrial waste. This, by growing in presence of this type of mixture of different industrial waste, this type of bacteria, they are giving knowledge to the researchers that they are able to growing in presence of complex hydrocarbons and complex waste which are present into the industrial waste. They can metabolize certain complex substances as well as they are able to collect the nutrient substances which are responsible for growth of different types of bacteria. This is the main significance of study of normal growth curve. Now move on to our topic. Our topic is normal growth curve, study of normal growth curve. From the BSc semester 3 microbiology paper 201, microbial physiology, unit 4, microbial growth, from Gujarat University syllabus. First of all, what is normal growth curve? When once bacteria are inoculated for growth into the fresh medium, they are allowed to grow for optimum period of time, and they are showing certain characteristic pattern of growth that is known as normal growth curve. Two factors, batch culture and closed culture system, are very important. Once bacteria inoculated into the fresh medium, they are they are allowed to complete their whole growth cycle into the same medium. That is known as batch culture or closed culture system. Now the different stages of growth curve. First is late phase or initial stationary phase. Second one is log phase or exponential phase. Third phase is maximum stationary phase. And fourth phase is death phase. When we are study the whole four phases of normal growth curve and plot the graph for normal growth curve, it is sigmoidal, S shape into the size. It is showing the slope of S. We can easily observe that S-shaped slope of normal growth curve. As I have mentioned, four different phases of normal growth curve. First is lag phase, that is the initial phase also. Second is log phase, also it is known as exponential phase. Third one is stationary phase. Here the death rate of cells and the growing cells, they are equal. Fourth is death phase of growth. Now first is lag phase. Lag phase, that is also known as initial phase of growth. In this phase, the bacteria, they are allowed to grow into the fresh medium. They are once inoculated in the fresh medium. In the fresh medium, they are allowed to grow for maximum period of time. Here in this phase, there is no visible increase into the cell number. Only the cell mass increases in this phase. Hence, this phase, it is not considered as idle phase, because in this phase, the number of cells are not increased, but cellular mass. Cellular mass, which type of cellular component? DNA, RNA, ribosomes and protein. That type of cellular mass that increases, and that allowed to grow the cell in their cellular mass. In this phase, the important parameters are the culture, which is we are inoculated into the new fresh medium. That culture must be taken from fresh culture medium, right? If it is obtained from death phase, then it is showing longer lag phase. And if the culture, it is containing different sporulating bacteria which are from the death phase, then that type of culture showing the longer lag phase. And the another component, another parameter which is affecting the lag phase, that is the composition of culture medium in which we are inoculating the culture. If that composition of culture medium is different than the previous one culture, then the bacteria, they are taking more time for their growth. The importance of lag phase, that is, as I have mentioned before, the bacterial cell size only increases, the number of cells are not increasing in this phase. The factors affecting length of lag phase, first is type of inoculum. Once the bacteria, they are taken from the old culture, that culture must be from the log phase. If that culture is from log phase, then bacteria, they are showing the faster growth rate. Why? Because in the log phase, bacterial metabolic activities are at the higher phase, right? That's why they are showing the maximum increase in the cell number, right? That's why the type of inoculum, it is the one parameter that affecting the lag phase. That if it is from log phase, then the bacteria are showing the shorter lag phase. Second parameter is if the inoculum, it is obtained from death phase, then it is showing the longer lag phase. That's why the inoculum must be from the log phase. Third parameter, if the culture containing the sporulating bacteria, then the sporulating bacteria, they are taking more time to convert to the vegetative cells. And they are, first of all, they are performing the whole sporulation cycle, and then the vegetative cells are produced. This is taking the more time for finishing of the lag phase. That's why the culture must be fresh, and must be, that is preferred, must be from the log phase, right? The second parameter is medium composition. The medium composition of the bacteria is rich in the nutrients, and if it is same of the previous culture medium in which the bacteria were growing, then it is showing the shorter lag phase, and the cell numbers are rapidly increasing into the log phase. Third one is size of inoculum. The optimum amount is must be required for the growth completion of the growth cycle, right? If the inoculum size is smaller, then it will take more time for completion of lag phase. That's why the optimum amount of inoculum must be required for shorter leg phase. In the type of inoculum phase, we can easily observe the step up and step down process in this phase. Second is log phase. The log phase is the rapidly growing period and bacteria are metabolically and physiologically active in the log phase. In the log phase, bacteria, they are dividing into the cell number by binary fission, as I have binary fission is discussed in our previous lecture topic. In the binary fission, bacterial number, cell number, they are increasing in the duplication. That's why the bacterial cell number increases rapidly, more and more rapidly. When the bacteria, they are starting to grow into the log phase, they are needing more amount of nutrients for their growth. Their metabolic activity is at higher rate and they are consuming more amount of available oxygen also, right? The different parameters that are affecting the log phase and the chemical and physical parameters as well as the nutrient parameters that are affecting the log phase. First of them all is, first is when the bacteria, they are metabolically growing into the one single fresh medium. After finishing of the leg phase, the metabolically activity is higher. Because of higher metabolically activity, the requirement of nutrients and requirement of available dissolved oxygen, that must be always higher, right? That's why the depletion of the nutrients and depletion of available oxygen is observed in this phase. The second parameter that affecting the log phase, that is the, if the cell numbers are increases rapidly, that's why because of increasing into the cell number, the viscosity of medium, nutrient medium which is available, that is also increases. Because increasing the viscosity of the medium, the availability of dissolved oxygen and nutrients becomes depleted. Third parameter which is affecting the cell number increase into the log phase, that is third parameter, is because of the constant metabolical activity, the respiratory oxygen, that is the demand for the bacteria, that is also increases. Because of that, the pH of the medium, that is decreased. Because of imbalance into the pH, that is also affecting the growth of bacteria, that also affecting the cell division activity. The fourth parameter is because of rapid increase into the cell number, the available space for growth of bacteria, that becomes insufficient. Because of increasing of bacterial cell number at higher rate, the space for movement of bacteria for growth, that become the compact space. That is, if it is the space for growth of bacterial cell number division, that is not enough, then the growth of bacteria become limited. And the last, most important parameter that affecting the log phase, that is because of respiratory cellular activity, the toxic waste metabolites, they are produced at the higher rate in the end of the log phase. Because of that, the cellular activity becomes lower and cell number, cell division, that becomes less. These are all about for the log phase. In short, in the log phase, the cell numbers, they are increasing rapidly and the metabolical activity and respiratory activity of the bacteria are higher. Third phase is stationary phase. After end of the log phase in the same medium, the depletion of the nutrients and availability of respiratory oxygen becomes lower At this in this phase, the number of the number of increase into the cells because of cell division that becomes lower comparative to log phase. The metabolical activity of surviving cells shows down, which stabilizes the population. In this phase, the exhaustion of nutrients and accumulation of waste product and harmful changes into the pH that affect the end of the exponential phase. The factors affecting stationary phase, they are the first important factor that affecting the stationary phase, that affecting the cell division, that is nutrient limitation. Because of increase of cell number excess into the log phase, the nutrients present into the medium that become in the less amount, that become exhausted. Second, limited oxygen availability, as we have studied into the log phase. Because of respiratory activity, the dissolved oxygen level into the medium that becomes lower. Third one is critical population density. Because of increase into the cell number rapidly, the free space available for movement of cells during the cell division that becomes lower, and the viscosity of the medium increases, the pH of the medium that become drop. Because of all the metabolic and physiological activities, the toxic waste products, they are increasing into the end of the stationary phase in the higher amount. Because of that, the growth rate of the bacteria that becomes equal to the death rate of the cell number into the normal growth curve, and at the end of the stationary phase of growth curve. Now last, fourth, death phase. In the death phase, all the cellular activity that becomes slower, all the cellular activity because of slowing of that, all the cellular activity, the number of cell division that stops, that near to stop because of exhaust of all the nutrients as well as the dissolved oxygen availability. These are the characteristic of the death phase. First is depletion of the nutrients. Second is waste material accumulation, loss of the ability of reproduction, resistance of the bacteria that show the extent survival. The death number of the cells can be logarithmic, and that is affected by the growth medium and culture. The main two parameters that affect the death phase, they are depletion of the nutrients, and second one is excess amount of toxic waste products. All the bacteria, they are not showing the same death phase. Some species of bacteria, for example gram-negative bacteria, they are showing the rapid decrease into the cell number, and they are showing the faster death phase. In the gram-positive bacteria, they are showing the slower death phase. The number of cells decrease slowly into the death phase in case of gram-positive bacteria. This is all about the normal growth curve. I have mentioned you 4 phases of normal growth curve: lake phase, log phase, stationary phase, and death phase. Here in the lake phase, inoculation of the medium, and the bacteria, they are try to survive into they are try to adapt into the new medium. In the exponential growth phase or log phase, bacteria rapidly increases, many living cells rapidly increases. In the stationary phase, that is near to death phase, number of cells decreases, few of the living cells are present, and the death rate of bacteria and growth rate, they are generally similar into the stationary phase. And the last, death phase, Number of cells are decreases, some cells are viable. This is all about normal growth curve. These are the references I have used for the topic. Thank you.